Hi, listeners, and welcome to episode 124 of Cell and Gene, the podcast. I'm your host, Erin Harris. So, depending on when you're hearing this, Rare Disease Day 2026 has just passed, but the urgency to develop better treatments for rare genetic conditions continues year round, which is why I'm excited to be joined by Dr. Ed Kaye, CEO of Aurora Therapeutics, to talk about a new approach to gene editing that aims to make personalized medicine more scalable and more accessible. Aurora is starting with PKU, a metabolic disorder with more than 1,200 known mutations, and using CRISPR technology to design therapies that can target multiple variants through a shared platform. Ed walks us through how strategies like trials and mutation-specific guide RNAs and smarter manufacturing could help bring gene editing to more patients faster, and why focusing on both science and commercial viability is key to making these therapies sustainable. All right, I'm going to let you go so you can check out my conversation with Aurora Therapeutics, Dr. Edward Kaye. Enjoy. Hi, Ed. Thank you so much for joining me on Cell and Gene, the podcast. It's a pleasure to have you.
Well, it's very nice to meet you.
Good. All right, so tell us a little bit about Aurora Therapeutics and its mission.
Okay. So Aurora Therapeutics was really founded by Jennifer Doudna and Fyodor Urnov, and really with the purpose of trying to expand gene editing, CRISPR, to more and more patients. And so I came on just starting in October, and our focus is, how do we kind of translate the wonderful science that has been accomplished in the past few years with CRISPR, but how do we actually turn it into a medicine that more and more patients can have access to it? So our approach, I would say, is a little bit more pragmatic than I think what other people are doing. We don't look at this as a science experiment. We look at this as a way of really making medicines and getting it to as many patients as possible. So the way we're focused on it is to go after one disease initially, and the disease that we're working on is phenylketonuria, or PKU, which is one of the older inborn errors of metabolism that's very well known because it was really the first newborn screening test that was put on the market, and it actually started here in Massachusetts in 1961. Because it's such a well-known disease, one of the challenges for CRISPR, however, it's not one common mutation. It's not like sickle cell. So you can't just make one drug to take care of all the patients. You have to take many, 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 many drugs. So the question is, how do we do that? So what we're in the process of doing is trying to really expedite and utilize a lot of the work that has previously been done. So what we're doing is we're using kind of tried and true components that have already been in humans. So the LNPs, the liquid nanoparticles, which are the delivery vehicles, the base editors, and so a lot of the things that we will be using, we know should very likely work and should be effective in patients. We're also targeting the liver, which has been a much easier organ to target with CRISPR. And so again, trying to use things that have a high likelihood of success. We like PKU for several reasons. Is that there's very good animal data to suggest that if you can get 10% of the liver edited, you should have more than enough of the enzyme to really make a very, very significant improvement in the disease. And we know that also based on studies that have been done in humans. If you have a phenylalanine hydroxylase, that's the enzyme that we're trying to edit and make sure we have enough of. If you have over 10% of that, you'll never have symptoms of the disease. So that's one of the approaches. The other thing we like about the approach using PKU is we can go after some of the common mutations first. And we know that the first few mutations will cover about 50 to 60% of the entire population. That gives us the ability to really hone in on the manufacturing process to make sure that we have the cost of goods as low as possible and have optimized the system. And one of the things we're doing with manufacturing is trying to really focus on a fit for scale. So in most diseases and in most drugs, the big problem you have is when you're going from a clinical stage and from a trial to get to commercial stage, you have to ramp up the production extensively. we're actually trying to make sure that we don't make too much, but make just the right amount of drug based on the mutation. So it's a little bit more of a Goldilocks phenomenon, because you can't have excessive amounts of drug that's very expensive to make and then not use it, or
Sure.
you don't have the half-life that would support it. So our focus is, again, making the right amount of medicine based on the mutation and based on the frequency of that mutation. The other thing that we're capitalizing on is some of the work that's been done by some of our collaborators over at the Children's Hospital of Philadelphia, and that's discussions with the FDA about having what's often referred to as a basket trial. And what that means is you're actually testing a number of different mutations in one trial. And this is not typical for kind of our industry. I had done that once before at Sarepta, which was the first company to develop an exon skipping therapy for Duchenne muscular dystrophy. We actually did one study where we tested two separate drugs in two separate genetic populations, but they had the same entry criteria for the study. And it was a very successful way to kind of expedite the clinical trials and the regulatory approval. So what we're planning on doing is actually studying a number of mutations simultaneously. And the reason is that the clinical endpoint is going to be the same. It doesn't matter what the mutation is. And one of the things we've learned about, and I think the FDA has agreed to this, is that gene editing is gene editing. It doesn't matter what the gene is. You're doing the same process. And so by testing a number, so the only thing we're really varying in all of these the studies is really just the guide RNA. Most of the components, over 98% of the drug that we're using is going to be the same despite the mutation. So we know that the LNP is going to be the same. It's never going to vary. We're going to use the same base editor. And what we're going to do is just vary the last 20 nucleotides in the guide RNA, and that's going to be different for each mutation. So there's a lot of similarity. And then what we're planning to do is to use this approach, but the regulatory, the manufacturing approach, to very quickly get into patients and really then once we get those common mutations, then we could go down the line and get the more and more infrequent mutations. But we've already developed the system, and now it's not a huge lift to continue to add more and more mutations because we've developed the system. We know how to do it. We know how to manufacture, just for the right scale. And then we add on to the regulatory approval. And our plan is to do it by in vitro testing. Once you've demonstrated that these components are safe and it works, now you can very quickly, without having to do clinical studies, add more and more and more mutations. And that makes it more of a commercially viable approach. And so we think this is a really good approach to try to get gene editing to those rare patients, but do it in a way where you're building on the experience for the more common mutations and then using that as you go down the line and pick off the rare and rare mutations.
Excellent. Thank you for that detailed description. And we'll get much deeper into the weeds, into PKU and gene editing. But before we do, I want to walk it back a little bit. At the top of our discussion, you mentioned, of course, Jennifer Doudna, Fyodor Urnov, who Cell and Gene, the podcast listeners and Cell and Gene readers know of quite well. And of course, their pioneering work in CRISPR and clinical translation. And so my question for you is, how has that scientific legacy shaped your approach to developing that scalable model you're talking about for personalized gene editing? And what makes Aurora distinct from earlier or other CRISPR-focused biotech companies out there now?
So I think, use the somewhat hackneyed phrase, we're standing on the shoulders of giants. We're using all of the basic science information that, Jennifer Doudna started in her first publication in 2012, and Fyodor has been working on gene editing, long before he came to Berkeley. And so we're using that, and Fyodor and Jennifer continued to advise the company. Fyodor is as a member of the board. So we have the ability to ask questions and get their advice, very often. And so that helps us a great deal. I think what makes us unique is, if you look at a lot of the history of gene editing programs, it's a little bit like I have this, really interesting idea, but I have all these shiny new toys, and I want to use this, and I want to look at all the shiny new toys. I think we've gone past that stage, and in my experience, being in the pharmaceutical industry, for almost 25 years now, you have to focus and you have to get that first therapy across the finish line. You have to show that it works. You can't be distracted by, oh, wouldn't it be interesting? We could do that too. And oh, we'll do that. But you have to focus. So I think for us, going after PKU, going after inborn errors of metabolism, we are hyper focused just on that. And what we're trying to do is really develop a commercial structure to use that science, but make it so we can actually have a commercially viable structure that allows delivering, drug and therapies and medicines to patients who need it, and do it in a much more cost-effective way.
Good. Okay. Earlier, too, you mentioned that one of Aurora's missions is to address the limitations of one-off treatments for rare mutations. So talk to us a little bit about the key economic, operational challenges, barriers, what have you, that have made these therapies unsustainable to date, and how is Aurora's platform approach really designed to combat that and overcome it?
Yeah, I think the two things that have really added to the cost of development, certainly one has been the manufacturing, the cost of goods, and how do you do it in a way. And there's been some great advances in that field. And what we're working with the contract manufacturers is to figure out ways to very efficiently produce smaller scale guide RNAs, and which is one of the most costly elements of that. So we think we can get down to a very reasonable cost of goods, so it doesn't suddenly present a sticker shock to everyone. I think the other concern is that there's, you can't do standard drug development and have an NDA or a BLA for every single mutation. That is not viable. And that's kind of what was done before. It's almost like we've taken the single drug approach and we've tried to make it fit for CRISPR. Well, it really doesn't fit because it's not one drug that we're trying to go after. I mean, again, as I mentioned previously, sickle cell, in some diseases, you are lucky. You have a common mutation that really all of the patients have. But that's the exception. That's not the rule. So how do we take care of the more common problem where you have lots of mutations? And how do you do it in a way? Well, one of the ways is that you can't do a study on every single patient. You have to be able to extrapolate and to be able to say, if we do this, the gene editing should be what we learn from one drug should apply to many, many, many therapies. And that's what I think what we're trying to do is to really set the precedent. So we're hyper-focused on PKU. We're using the regulatory precedents that have been set. We're using the advances in manufacturing, and we're coming up with therapies very quickly to get into patients. And we do it in a way where if you're already treating 50 or 60% of the population, you're making the revenue. But then that allows you then to add on more and more and more patients, which continues to increase the revenue, but at a fraction of the cost.
Sure. One of the many interesting and important aspects of Aurora that made me want to talk to you and, just anyone on your team is that how you and your team plan to leverage the FDA's new plausible mechanism pathway, which represents a major regulatory shift for gene editing therapies. So talk to us a little bit about what that pathway enables and how it's changing your development strategy for programs like PKU.
Yeah. So one of the things that's important to remember is, even going back a number of years, there are pathways in the FDA for more accelerated approvals. Now, I've used that many, many times in the past. I've used it when I was at Genzyme, where, again, you could use a surrogate endpoint, which would be the accelerated pathway, where you have an endpoint which is likely to predict a clinical benefit. And so that mechanism has really been there, I think, at the FDA for a time. The one thing that's interesting for us with PKU is the endpoint of the phenylalanine level in the blood is actually a clinical endpoint, a hard clinical endpoint that has been used for approval for other drugs in PKU. So we're a little bit ahead, I would say, of let's say that other indications, because we don't even have to come up with a plausible mechanism. We have a mechanism that has already been really demonstrated to have full clinical approval using this hard clinical endpoint. But I do think what we're seeing in the FDA is, and I do feel that it's quite sincere, a lot more flexibility for the rare disease population. We've seen this for the manufacturing. And I would say the best precedent, even more so, which is part of the, I guess, of the plausible mechanism pathway, is what we've learned from some of our collaborators. And we know that the group over at the Children's Hospital of Philadelphia have had discussions with the FDA. They've been allowed to do a study, an umbrella study, looking at 6 different mutations simultaneously. They've also had the discussion of looking at this at urea cycle disorders, where you're actually measuring a number of different genes at the same time and doing gene editing, because the endpoints are pretty similar between these, but they're actually different genes in the same pathway. And so I think those are the precedents that we plan to use is, we have to have something different for CRISPR in order to get to all the patients. It cannot be the same regulatory pathway. And I think what we're seeing now, in the FDA is an appreciation that CRISPR is different and that gene editing shouldn't make a difference what gene you're editing. It's the same process. And so I think as we learn more and more and more, and working with the regulatory authorities, I think we'll be able to do faster regulatory approvals, because it's really the manufacturing costs and the regulatory costs and the time it takes that causes it to be so expensive. And one of the things too is we are going for liver targeted in vivo. I mean, one of the challenges, people always bring up Casgevy as a thing. Well, that's not commercially viable. And I would say it's too early. And I think one of the problems also is, I think that wasn't appreciated, and talking with some of the investigators who've been involved in that is, I don't think anybody quite figured out how difficult it is to get cells. You have to do an apheresis. In other words, you have to basically filter out certain blood cells, white blood cells, and capture them. And sometimes it takes 3 different times to get enough cells to put it back into the patient with the CRISPR. So there are I think some issues that are unfairly suggesting that CRISPR is not viable. I would say it is viable. It's a very reasonable approach. And if you think about it, if you can have a one-time therapy that I'll use the word cure. Everybody always is a little hesitant to use it. But if you could do that, if you think there could be almost nothing that's more cost effective. Because if you think of some of the current therapies for rare disease now, you're talking, $400,000, $500,000, $600,000 a year upon year upon year. If you could have a single therapy or a series of therapies where you don't have to treat the patient forever and you have a really, really good outcome, that can be very cost effective. So I think, what we're trying to do is make sure that we people know that this could from a health economics outcome, this could be, very positive, and even thinking of insurers, because you don't have cost year after year after year. It's a cost-effective way of really trying to improve health in America.
Yes, sure. At the top of our call, I mentioned that we're going to get more into the weeds with PKU, and so that's what I want to talk about next. We know that it's a genetic disorder with over 1,200 known mutations. What's the scientific and clinical rationale for Aurora to start with PKU, and how will your editing platform adapt over time to address any additional mutations in PKU and then beyond?
Yes. So PKU has a number of advantages, and I think first of all, it's a fairly large population, given that it is a rare disease. And we know there's about 15,000 classical PKU patients in the United States alone. So that's a large population to deal with. And one of the challenges, of course, in rare disease is if it's going to take you forever to find the patients to do a study, that's not going to be very, easy to do. It's going to be challenging, and and obviously it increases the cost. So we have a population. We also have a population that currently still continues to need a better treatment. So really the foundation for treatment in PKU, and this is beginning in the early 60s, has been the diet. So you're reducing the amount of phenylalanine in the patient's diet so that you don't have very high levels of phenylalanine that we know are toxic to the nervous system. The problem is that works very well if you're an infant on formula, and you can have a very specific formula. You know exactly how much phenylalanine you're getting, and you can control the amount of phenylalanine that patients are receiving. The challenge we have is once you get to be an older child and an adolescent and an adult, phenylalanine is so ubiquitous in the food supply. It's in everything. And so what we realized, it has been really challenging to maintain a diet, especially in older children and adults, to keep that phenylalanine at a level below where it becomes toxic. And there was a recent study also out of the Children's Hospital of Philadelphia where they were just following their patients and very compliant families and patients. But what they realize is that the majority of the time, the patients are not under good control, and they come into the clinic and their levels are in the toxic range, despite being on the diet. So that means that we really do need better therapies. And I think, some of the therapies have been primarily um, cofactors like tetrahydrobiopterin, and that in some of the milder mutations can help increase the enzyme activity, but it's never enough to really make a huge difference in the patients. It's an incremental difference. And there's also, been tried to replace the enzyme, give it an infusion, but you have to give it every day. And again, the half-life is quite small. But so by doing gene editing, you're really restoring the production of the phenylalanine hydroxylase in the liver, and you're allowing the liver to correct for the deficiency. So it's a much more, let's say, basically it follows the pathways of the metabolism within the body, and you're allowing the body to kind of restore that normal function rather than trying to replace it extraneously. And so it's a good system. I think the other thing that we liked about from a PKU standpoint is there's a regulatory precedent so that if you we know that thresholds of 360 micromoles per liter is an approvable endpoint, and we in the U.S. And it's a 600 micromole threshold that's seen in Europe. So again, there's a regulatory precedent that demonstrates that if you can reduce that phenylalanine level, that's enough to get the registration for the product. So, and that also means that it's a fairly short study. We know within days, if it's effective, you're going to see a drop in that phenylalanine level based on the animal model. So you can do a fairly efficient, quick study, not terribly difficult for patients to comply to, and you'll have an answer. And so those are one of the reasons why it's really the knowledge about the disease, about the regulatory precedent, and about the rapidity of how quick this therapy will work if it's effective.
Sure. Now, before I let you go, at the top of the call, and of course, we know that you mentioned your work with Sarepta. We know you've worked with organizations like Stoke, and that really have redefined innovation and rare disease development. And so, based on your experiences there and throughout your career, how are those companies helping to influence how and what you're building from Aurora's culture's perspective and the partnerships you're working on in this next era of gene editing?
Yeah. So I was really fortunate. So my background is in pediatric neurology and biochemical genetics. So, I had firsthand experience of actually treating these patients and seeing some of the challenges of the treatment that we were developing. So I was fortunate to go to Genzyme for my first experience in the biotech industry. And one of the things that came natural to me, but that was very in the culture in Genzyme, was patients came first, and you understood that you needed to work with the patients. You needed to learn what was important to them, what did they need, and they were the ones that really helped you with the design of the study. They told you what was important. They helped, find more patients that would be part of your study. They worked with regulatory authorities. They worked with reimbursers to get the drug paid for. And then we were, Sarepta was an excellent example of working with the Duchenne muscular dystrophy organizations. And we would not have been able to get that drug approved if it wasn't for all of the wonderful work and the support that we had from the muscular dystrophy societies. And then taking that also, the first genetic therapy for epilepsy is very likely going to be coming from Stoke. And it really changed the way we think about epilepsy. It was upregulating the missing protein and trying to address the underlying genetic cause, not just treat the symptoms. And again, that was an example of the Dravet Society in the United States and in the UK and in Europe were incredibly supportive. And again, we could not have done that kind of work without the support of all of those groups. So I think, what we're doing is trying to make sure that we have the culture that everyone in the company, no matter what function they have, they know that patients come first, and that our mission is to supply drugs for the patients who need them. And then, Henry Termeer, who was the CEO of Genzyme, said, good medicine is good business. If you have a medicine that's really helping patients, that's good business. It'll be successful. And so as long as you keep that in the back of your mind and always make sure that you're addressing the needs of the patients. And one of the things I've certainly learned is that no matter how difficult your day is, it pales in comparison to the challenges that patients have with these genetic diseases. And whatever we can do to make their lives better is really important.
Wonderful. Well, I absolutely love that altruistic and thoughtful and patient-centric mission that you and your team at Aurora are putting forth into the work that you're doing. So thank you for that. And that brings us to the end of our episode featuring Dr. Ed Kaye, CEO of Aurora Therapeutics. So Ed, thank you so much for your time. This was a real pleasure and a treat to hear all of that's going on at Aurora. And we wish you the best as you're going forward, and love to have you back sometime soon to have any type of progress update.
I'd be happy to do that. It was very nice to be invited. Thank you very much.
Absolutely. All right, listeners, thanks for tuning in, and be sure to tell your peers to subscribe to Cell and Gene, the podcast, on any major podcast platform, and visit us at cellandgene.com. And we will talk to you soon.